Okay, let's unpack this. We are diving deep into, well, one of the most compelling and, genetically speaking, really asymmetrical areas of single gene disorders, X-linked inheritance.
Welcome aboard. Yeah, we've synthesized your source material today, and it centers intensely on these genetic conditions. Our mission, really, is straightforward:
Love
to
that.
give you, the listener, a comprehensive, crystalline view of X-linked disorders.
Right.
We need to understand precisely why these conditions affect males and females so drastically differently.
And how just one single gene mutation on that critical X chromosome can unleash these incredibly complex system-wide diseases.
Exactly, it's quite something.
The scale of genetic issues is often, well, surprising, isn't it? Our sources estimate that genetic disorders have a lifetime frequency of something like 670 per 1,000 people. That's huge.
It is, and within that massive category, you've got the single gene mutations, the classic Mendelian disorders forming a large subset,
Hmm.
and X-linked conditions are distinct because they are entirely defined by the unique and sometimes, frankly, brutal architecture of our sex chromosomes.
That's the key context then. Most of our genetics relies on those 22 pairs of, reliable autosomes, where genes usually come in compensating pairs.
Right, backup copies.
But the two sex chromosomes, the X and the Y, they introduce this profound asymmetry.
Absolutely. And that asymmetry fundamentally changes the rules of genetic expression. It creates a system where, well, males are inherently more vulnerable.
So let's start with the chromosomal players.
Mm.
Females typically have two large X chromosomes, XX. Well, males have one X and one, well, tiny Y chromosome, XY.
Right, and the sources are quick to point out that the Y chromosome is mostly inert when it comes to the vast catalog of human diseases.
Mostly inert, yeah.
It truly is. The Y chromosome is largely non-homologous to the X. It carries relatively few functional genes, and those genes it does carry are primarily related to, well, determining male sex development and, critically, spermatogenesis.
Fermatogenesis.
And this leads to a crucial genetic fact. If a male suffers a mutation affecting a gene on his Y chromosome, he is typically infertile.
Ah, right. And if he's
He
invert,
can't pass the trait on, exactly.
so no Y-linked diseases passed down generations.
Precisely. This means that true Y-linked inheritance, a disease passing reliably from father to son, is, statistically and clinically pretty much non-existent.
Okay.
Therefore, whether we're talking about recessive or dominant inheritance patterns, all clinically relevant sex-linked disorders are, in fact, X-linked.
That brings us right to the core concept, explaining why males are more vulnerable. Hemizygosity. Sounds complex.
It does, but it's really just, as you said, common sense based on that chromosome count.
Okay.
Hemizygosity is the mechanism. It explains why X-linked recessive disorders are predominantly expressed in males. Since a male possesses only one X chromosome in that non-compensating Y, he is hemizygous for every single gene on that X chromosome. He simply cannot rely on a paired normal allele on another X to mask a mutation.
It's like walking a tightrope without a net, genetically speaking.
That's a good way to put it. If the X chromosome he inherited from his mother carries a mutant gene, there is simply no backup copy to step in.
And the disorder is expressed fully, just like that.
Just like that. It's the genetic cliff edge, as you called it.
So to put this into perspective for you listening, think about an autosomal recessive disorder like cystic fibrosis.
Right. With CF, you typically need to inherit two copies of the mutant gene, one from each parent, to actually get the disease.
Okay.
But with these X-linked recessives, the male needs only one copy of the mutant gene. He gets it from his mother, and bam, the disease manifests.
That efficiency in disease manifestation, that's the hallmark of hemizygosity.
It really is.
Now, let's map out the transmission patterns. Understanding these pathways is, well, it's absolutely vital, isn't it? Particularly for genetic counseling. The risk patterns are so specific.
They are. And we always start by analyzing the path from the affected individual. So if we consider an affected male, a father who manifested the disorder, he possesses that mutant X.
Right.
Now, when he reproduces, he transmits his Y chromosome to all his sons.
Right. The Y determines maleness.
Correct. So the famous rule emerges. An affected male cannot transmit an X-linked condition to his sons.
Cannot. That's a critical rule. Helps distinguish X-linked from autosomal conditions.
Absolutely. Cannot pass it to sons. However, all of his daughters receive his mutant X chromosome.
His only X chromosome.
His only X. Therefore, all his daughters will be obligate carriers.
Obligate carriers, meaning they must carry the gene.
Yes. Barring very rare exceptions, they generally won't express the full disease themselves, but they carry that genetic liability forward.
Which makes the daughters the crucial link for the next generation. So let's focus on that heterozygous female carrier, maybe the daughter of that affected father, or perhaps a woman whose own mother was a known carrier. What's the risk she passes the trait on to her children?
Right. This is the critical 50% risk scenario. Since she has one mutant X and one normal X, there's a 50% chance for each child she has, regardless of their sex, to inherit the mutant allele.
Okay, 50-50 chance for any child.
Yes. Now, if she transmits that mutant X to a son...
He only gets that one X.
Exactly. Being hemizygous, he will manifest the disorder. So sons of heterozygous women have a 1 in 2 or 50% chance of developing the condition.
50% chance for each son.
Wow.
And if she passes the mutant X to a daughter?
Then that daughter has a 50% chance of being a carrier, just like her mother.
But she typically remains phenotypically normal.
Typically, yes, because she still has that paired normal allele on her other X chromosome.
Okay.
It's this predictable 50% risk to the male offspring of female carriers that makes X-linked recessive inheritance such a distinct and well-defined pattern within Mendelian disorders.
It really drives the pedigree analysis for families at risk, as our sources stress.
Absolutely. It's the core mechanism.
Okay, moving on to the female carriers. We've established they usually don't express the full condition because they have that paired normal allele. But, you know, their status isn't just passive protection. It's a dynamic biological state.
It really is, defined by a fascinating cellular mechanism.
And here's where it gets really interesting, right? X chromosome inactivation.
Yes, or lionization, as we call it, named after the geneticist Mary Lyon. This process is the primary reason women are protected from the full severity of X-linked recessive disorders.
But it's also why the line between carrier and affected can get, well, blurry.
Exactly. It introduces variability.
So tell us why this process happens in the first place. Why does the body randomly choose to just shut down an entire X chromosome?
It's all about gene dosage compensation. Think about it. Males only have one X chromosome's worth of genes active. Females have 2 X's.
Right.
If females expressed all the genes on both X chromosomes, they'd effectively have double the protein product for thousands of genes compared to males. That would cause major physiological imbalances.
Okay, so it's about leveling the playing field between sexes.
Precisely.
Yeah.
To equalize this dose, very early in female embryonic development, one of the 2 X chromosomes, it could be the one from her father or the one from her mother, is randomly and permanently inactivated in every single somatic cell.
Permanently inactivated. Wow. So every cell essentially flips a coin, deciding which X gets shut down for the rest of its life.
That's a great analogy. It flips a coin. The inactive X then condenses down into a tiny, dense structure called the Barr body.
A Barr body?
You can actually see it under a microscope. Think of it like that X chromosome being, I don't know, shoved into a closet, locked down, and functionally ignored by the cell's machinery.
Okay.
And because this decision, which X gets inactivated, is random in each cell lineage early on.
The female body becomes a mosaic.
Exactly. A mosaic of 2 distinct cell populations.
Meaning some cells are running on the normal X and some cells are running on the mutant X.
Yes. They have a variable proportion of cells where the mutant X chromosome is active, producing the defective protein, or no protein, and a variable proportion where the normal X chromosome is active, producing the functional protein. This mosaicism explains their partial protection, usually. And crucially, it also explains their variable expressivity. Most women will have a ratio somewhere near 50.50 between the 2 cell types.
Which means they produce enough normal protein overall to stay healthy.
Generally, yes. But our sources were clear. We absolutely cannot rule out expression entirely in females. This protection is not absolute.
Right.
That's where the nuance lies. If the inactivation isn't perfectly random, if, just by chance, the normal allele happens to be the one inactivated in a disproportionately large number of cells.
And the mutant allele is the one that's active more often than not.
Precisely. In that case, the female could express the X-linked condition fully, or at least near fully. This is known as skewed lionization, or skewed X inactivation.
Skewed lionization. It effectively unmasks these recessive traits in females who are only expected to be carriers.
It does. It happens.
We definitely need a solid example of this mosaicism in action. The sources highlight glucose-6-phosphate dehydrogenase deficiency, G6PD deficiency, as a perfect case study here.
Yes, G6PD deficiency is a classic. It's transmitted on the X chromosome, and like other X-linked recessives, it's expressed principally in hemizygous males.
Right. causes acute hemolytic anemia when triggered.
Correct. Now, for the heterozygous female, because of that random X inactivation, a certain proportion of her red blood cell precursors will have silenced the normal X allele, the one carrying the functional G6PD gene.
So those specific red blood cells are genetically defective. They lack the protective G6PD enzyme.
Precisely. And these defective red cells are highly susceptible to damage from external factors that induce oxidative stress.
Things like certain foods. Fava beans are famous for this, right?
Fava beans, yes. And also exposure to certain medications like the anti-malarial drug Primakine.
Okay.
So the female isn't just a carrier of the trait in her DNA, she is also potentially susceptible to these drug-induced hemolytic reactions herself.
What really stands out to me here, though, is that it's not usually an all-or-nothing situation for her. The susceptibility is typically milder than an infected male.
Yes, and that comes back directly to the mosaicism. Because of the random nature of inactivation, the proportion of defective red cells in heterozygous women is almost always less than 100%.
So if, say, 70% of her red cells are normal,
She might show only a mild reaction, perhaps only under severe oxidative stress. But
if she had that skewed lionization we talked about, maybe 85% of her cells lack normal G6PD.
then her reaction would be much closer to that of an affected hemizygous male. It really reinforces the idea that X inactivation creates a spectrum of outcomes, not an absolute barrier.
And this ties directly back into pharmacogenetics, doesn't it?
Yeah.
The study of genetically determined adverse reactions to drugs.
Absolutely. This enzyme deficiency is a textbook example of a hidden genetic trait that's suddenly unmasked by an environmental or chemical factor like a specific drug.
It really shows that even a sophisticated protective mechanism like lionization has its limits. It offers partial protection, maybe, rather than absolute immunity.
Exactly. It makes that distinction between carrier and affected very blurry indeed in some cases.
Okay, so the list of known X-linked recessive disorders is, well, it's vast. And what they often share is this remarkable pleiotropism, right?
Yes, pleiotropism is a key concept. A single mutation on the X chromosome can lead to defects across multiple, seemingly unrelated body systems.
Our source material lists major categories affected: musculoskeletal, blood, immune system, metabolic pathways, nervous system.
It's a wide range.
Yeah.
Let's delve into the mechanics of a few of the most classic examples to see how this works.
We should probably start with the musculoskeletal system, specifically Duchenne muscular dystrophy, or DMD. This is a condition that really illustrates the catastrophic failure of just one crucial structural protein.
DMD is truly devastating. It's a progressive muscle wasting disease, and it almost exclusively affects boys. The underlying cause is a mutation in the DMD gene.
Which is located on the X chromosome.
Yes, and what's fascinating, or perhaps daunting about this gene, is its sheer size. It is the largest known human gene, spanning about 2.4 million base pairs of DNA.
2.4 million. Why does the size of the gene matter so much?
Well, think of it like this. A larger target is easier to hit. A larger gene offers a larger area for spontaneous mutations to occur just by chance during DNA replication or repair.
Ah, okay. So that contributes to why DMD isn't incredibly rare, despite being so severe.
Exactly. That high spontaneous mutation rate is a factor. Now, the DMD gene itself provides the instructions for making a protein called dystrophin.
Dystrophin. And what does dystrophin actually do in the body?
Dystrophin acts as a crucial structural anchor, kind of like a shock absorber, within muscle fibers. It links the muscle cell's internal cytoskeleton, its scaffolding, to the extracellular matrix, the environment outside the cell.
So it provides stability, especially during muscle contraction.
Precisely. It helps maintain the integrity of the muscle cell membrane during the stress of contraction and relaxation.
So if dystrophin is missing or severely dysfunctional, like in DMD, what happens?
Without that essential anchor, the muscle membrane becomes fragile and leaky. Repeated contractions cause microtears.
Ouch.
Yeah. This leads to chronic inflammation, muscle cell death, and eventually the replacement of functional muscle tissue with fat and fibrotic scar tissue.
And that explains the progressive weakness, starting usually in the large muscles of the legs and pelvis.
Yes. But the damage is widely pleiotropic, as you mentioned earlier.
Right, pleiotropism again. The sources remind us the consequences aren't just limited to walking ability.
That's absolutely right. Dystrophin isn't just in skeletal muscle, it's also essential for the structural integrity of the cardiac muscle, the heart.
So heart problems are common too.
Very common and serious. Cardiomyopathy, a weakening and often enlargement of the heart muscle, is a major cause of mortality in individuals with DMD.
And the brain.
And the brain. Furthermore, certain dystrophin isoforms, different versions of the protein, are crucial in brain tissue. This means that cognitive impairment and learning disabilities can also be part of the syndrome.
All stemming from that single, missing, or non-functional X-linked protein. Incredible.
It really illustrates the principle.
Okay, so we've seen how X-linked mutations can destroy muscle structure. They can be equally devastating in the blood system. That brings us to the classic disorders, hemophilia A and hemophilia B.
Yes, hemophilia, the bleeding disorder. It's perhaps the most historically famous X-linked condition, isn't it?
Yeah, largely because of its presence in European royal families. That actually helped illuminate the inheritance pattern long before we understood the molecular genetics.
It did, a sort of tragic natural experiment in pedigree analysis.
So what's the molecular difference between hemophilia A and B? They both cause bleeding problems.
They do, but they stem from defects in different parts of the clotting process. Both involve deficiencies in specific clotting factors, which are proteins needed for blood to clot properly. Hemophilia A results from a deficiency in clotting factor 8.
Factor 8.
While hemophilia B results from a deficiency in factor X, factor 9.
And both of these genes for factor 8 and factor 9 are located on the X chromosome.
Correct. And both factors are absolutely essential components of the intrinsic pathway of the blood coagulation cascade. Think of it as a complex chain reaction.
Okay, so if a male is hemizygous for a mutation affecting either factor 8 or factor 9X, what's the consequence?
The blood simply cannot clot effectively. While, minor cuts and scrapes might eventually start bleeding, the real danger lies elsewhere.
Right.
It's the spontaneous bleeding episodes, often into joints, we call that hemarthrosis, and into muscles. This internal bleeding can cause chronic, severe pain and debilitating permanent joint damage over time.
And there's a risk of more severe bleeding, too.
Absolutely. The risk of life-threatening hemorrhage, particularly internal bleeding in the abdomen, or, most dangerously, intracranial bleeding inside the skull, is ever present, especially after trauma, but sometimes spontaneously.
The high predictability of this X-linked pattern, though, means that once hemophilia is identified in a family, genetic counseling becomes highly precise, right? Regarding that 50% risk to male offspring of female carriers.
Extremely precise. It's a textbook example of Mendelian inheritance in action, driving clinical practice.
Okay. Moving now from structural proteins and clotting factors to the nervous system, let's explore Fragile X syndrome. This one introduces really fascinating instability into the whole genetic process.
It really does. Fragile X syndrome, or FXS, is known as the most common inherited cause of intellectual disability and also a common cause of autism spectrum disorder.
And our sources stress that it doesn't follow strict Mendelian rules. Why not?
Because it results from a specialized, rather unusual type of mutation. The trinucleotide repeat mutation.
Trinucleotide repeat. Okay, break that down.
This is a truly dynamic genetic anomaly. It involves the unstable amplification, like a stutter in the DNA sequence, of a short sequence of 3 nucleotides, specifically the sequence cytosine guanine guanine, or CGG.
CGG.
This CGG sequence is located within the regulatory region, kind of like the on-off switch of a gene called FMR1, which is on the X chromosome.
Okay, and in a normal person?
In the general population, the average number of these CGG repeats is relatively small, maybe around 29 or 30 repeats.
But in Fragile X syndrome, that sequence dramatically expands. It gets much, much longer.
That's the dynamic part. It's unstable. In affected individuals, the repeat count explodes. It amplifies from, say, over 200 repeats up to potentially 4,000 tandem repeats, just CGG over and over again.
4,000, that's a huge expansion.
Yeah.
What does that massive block of repeats do to the gene?
It causes the regulatory region of the FMR1 gene to become chemically modified through a process called methylation. Think of it like sticky tags being added to the DNA.
And methylation usually silences genes, right?
Exactly. This hypermethylation completely shuts down or silences the FMR1 gene.
So silencing the FMR1 gene means no functional FMR1 protein is produced.
Correct. No protein or very little. And the FMR1 protein, also called FMRP, is vital for normal brain development, particularly for synaptic function, how nerve cells communicate, and synaptic plasticity, which is crucial for learning and memory.
So its absence leads directly to the core features of the syndrome, the intellectual disability, often accompanied by characteristic behavioral issues like anxiety, hyperactivity, and autistic features, and sometimes physical features too.
Yes, that's the link.
And the really distinguishing feature here, you said it was dynamic, the concept of anticipation. The mutation is unstable.
Precisely. The mutation isn't static from one generation to the next. The degree of amplification, the number of CGG repeats, tends to increase, particularly during female gametogenesis when eggs are formed.
So when a female carrier passes it on,
Yes. A mother might carry what's called a premutation, say somewhere between 55 and 200 repeats. This premutation range often doesn't silence the gene completely, but it makes that repeat sequence highly unstable.
go.
When she passes that gene, that unstable premutation, to her child, the repeat count can amplify significantly, sometimes massively, crossing the threshold into the full mutation range, over 200 repeats.
Which then leads to the gene silencing and the manifestation of Fragile X syndrome in her child, especially sons.
Exactly. This explains why the disorder sometimes appears to become more severe or have an earlier onset in successive generations within a family, a phenomenon known as anticipation.
That concept that our own reproductive cells can actually change the genetic information we pass on is quite humbling, isn't it?
It really is. It shows that even seemingly straightforward Mendelian inheritance can be complicated by these unstable, dynamic genetic elements. It adds another layer of complexity.
Okay, one more example from the sources. Let's pivot to a purely metabolic condition, Lesch-Nyhan syndrome. This one really exemplifies how an enzyme defect can lead to devastating system-wide injury.
Yes, Lesch-Nyhan is a profound example of metabolic disruption.
And it's not just from a missing product, but from a toxic buildup of intermediates.
Yeah, it's a key part of it, yes. Let's walk through the biochemical mechanism.
Please do.
Lesch-Nyhan syndrome results from a severe, near-total deficiency of an enzyme called hypoxanthine guanine phosphoribosyltransferase, or HGPRT.
HGPRT.
Got it. This enzyme is crucial in what's called the salvage pathway of purine metabolism. Purines are essential building blocks for DNA and RNA. The salvage pathway essentially recycles purine bases so the body doesn't have to constantly build them from scratch, which is energetically expensive.
Okay, so HGPRT is a recycling enzyme for purines. Why does the lack of it cause such profound neurological issues and, well, the characteristic and distressing self-mutilation often seen in this syndrome? That seems like a huge leap.
It does seem like a leap, and the full connection isn't perfectly understood, but a major part of it is due to a failed system of feedback inhibition.
Feedback inhibition.
Yeah. Normally, the end products of a metabolic pathway act as signals. They provide negative feedback to the early enzymes in the same pathway, telling them, okay, we have enough, slow down production.
Like a thermostat regulating temperature.
Exactly like that. Our sources explain that the deficiency of HGPRT prevents the proper formation or utilization of these crucial end products in the salvage pathway, when those end products are effectively missing from the feedback loop.
The brake on the main purine synthesis pathway is removed.
Precisely, the brake is off. The entire system for de novo purine synthesis, building purines from scratch, goes into massive, unregulated overdrive.
It just keeps making more and more.
More and more, generating huge amounts of intermediates and also catabolic products, breakdown products from all those excess purines. One major product that builds up is uric acid.
Ah, uric acid, like in gout.
Exactly. High concentrations of uric acid build up in the blood and tissues, leading to severe gout-like symptoms, kidney stones, and potential kidney failure. That's the metabolic consequence.
But the metabolic catastrophe clearly goes far beyond just uric acid accumulation, impacting the brain so severely.
It absolutely does. While, as I said, the full mechanism linking the purine defect to the neurological symptoms is still debated, it's clear that the absence of HGPRT and the resulting accumulation of other purine intermediates or altered neurotransmitter levels leads to severe neurological dysfunction.
Which manifests as?
It manifests as involuntary movements, muscle spasticity, dystonia, cognitive impairment, and that highly characteristic and distressing compulsion towards self-injurious behavior, like biting fingers and lips.
The complexity here is really striking. The missing enzyme isn't just causing a local lack of something, because HGPRT is needed pretty much everywhere in the body.
Yeah.
The defect disrupts the entire metabolic balance, leading to these wide-ranging pleiotropic effects, metabolic, neurological, even hematological sometimes.
Yeah.
That connection is absolutely powerful. Whether it's a failure to properly construct a structural protein like dystrophin and DMD, a failure to regulate the blood clotting cascade in hemophilia,
Right.
or a failure to regulate a complex metabolic pathway, removing crucial feedback control, as in Lesch-Nyhan. These X-linked disorders often result in devastating, wide-ranging effects, amplified by that state of hemizygosity in males.
Now, while the majority of the clinically significant X-linked disorders we hear about are recessive, like the ones we've just discussed,
Yes, the vast
we
majority.
absolutely must cover the rare but critical exceptions, X-linked dominant condition.
We must. They follow different rules.
So a dominant trait on the X chromosome,
Yeah.
that must change the expression dynamic significantly, right?
Yeah.
How does this shift the vulnerability between males and females compared to recessive?
Well, the fundamental rule of dominance still holds. A single copy of the mutant allele is sufficient to cause the disease.
Okay, one copy is enough.
One copy is enough. Now, because the female has 2 X chromosomes and the mutation is dominant, expression does occur even in heterozygous females. They will show the disease.
So females are affected in X-linked dominant inheritance.
Yes, they are typically affected. However, an interesting point is that the disease often presents less severely in females than in males.
Why is that?
Again, it likely comes back to lionization, X inactivation. Even though the mutation is dominant, the random inactivation means that roughly half of her cells are expressing the normal allele from the healthy X chromosome. This mosaicism, while not preventing the disease entirely, often tempers the overall severity compared to a male who only has the mutant allele active in all his cells.
Ah, okay, so lionization still plays a role, just a modifying one.
Exactly, a modifying role. The key difference, then, isn't so much whether the female is affected, she usually is, but rather how the disease is transmitted, particularly by an affected father.
Right. The transmission patterns are what really distinguish X-linked dominant from both X-linked recessive and also from autosomal dominant conditions.
Precisely. Let's look the affected heterozygous female first. She has 1 mutant X and 1 normal X. So she still transmits the disease to, on average, half her sons and half her daughters.
50-50 chance for each child, just like autosomal dominant.
Correct. Both sons and daughters have a 50% chance of getting her mutant X chromosome.
Okay. Now the hallmark distinction, the affected male. He only has 1 X chromosome, and it carries the dominant mutation.
Right. So if he is affected, he transmits that dominant X-linked allele to all of his daughters. 100%.
All daughters get his only X, so they will all be affected.
They will all be affected, yes, though, as we said, likely less severely than him. Critically, though, because his sons only receive his Y chromosome.
Assuming the mother is unaffected.
Assuming the mother is unaffected, yes, the affected male transmits the condition to none of his sons. 0%.
0% of sons. That is the definitive distinction, isn't it?
It absolutely is. If you see a pedigree, a family tree, where an affected father passes the condition to 100% of his daughters, but 0% of his sons, you are almost certainly looking at X-linked dominant inheritance.
And there are conditions that follow this pattern.
Yes, though they are rarer. Examples include vitamin D-resistant rickets, also known as X-linked hypophosphatemia, and some forms of Alport syndrome, which is a kidney disease often associated with hearing loss.
Okay. Wow, we've covered a tremendous amount of ground here. From the basic architecture of the sex chromosomes all the way to the intricate consequences of specific enzyme failures.
We really have. It's a complex area. Perhaps we should try and gather the, say, 3 essential takeaways from this deep dive into X-linked genetics.
Good idea. Let's crystallize the key points for everyone listening. Takeaway number 1.
Takeaway 1. X-linked disorders are fundamentally defined by their expression in hemizygous males. These males manifest the disease because they lack that normal compensatory allele on a second X chromosome.
Right. That hemizygosity makes them acutely vulnerable to any mutation carried on the single X chromosome they receive from their mother.
Perfectly put. Takeaway 2.
Takeaway 2. The disorders are primarily transmitted by heterozygous females, the carriers. They usually don't show the full disease but pass the trait onto their sons with that predictable and often worrying 50% risk.
Yes. This maternal transmission pattern is absolutely central to pedigree analysis and genetic counseling for these conditions.
Okay, and the third, and maybe the most complex takeaway.
The third takeaway has to be about the female carriers themselves. They are typically protected due to the random inactivation of 1 X chromosome lionization.
But that protection is conditional, isn't it?
It is conditional. The extent of this random inactivation, whether it's balanced or skewed, dictates their precise level of expression or susceptibility.
It demonstrates that their status is really a functional mosaic, not an absolute immunity. G6PD deficiencies show that so clearly, especially with the pharmacogenetic implications.
Exactly. It highlights that variability.
So if we connect this all back to the bigger picture,
Well, understanding the X-linked pattern is just critical, I think, because it forces us to look beyond simple dominant recessive categories. It makes us appreciate the sheer nuance of genetic compensation, dosage effects, and the intricate cellular regulation that goes on in human biology.
And this knowledge provides the basis for more precise genetic counseling, right?
Yeah.
It allows families to understand the specific risks involved in these complex conditions, whether it's the muscle weakness of DMD or the metabolic chaos of Lesch-Nyhan syndrome.
Precisely. It empowers understanding and decision-making.
Okay, so this raises an important final question, something for you, our listener, to maybe mull over. Given this fascinating phenomenon of random X inactivation, which acts as a sort of cellular regulator, and knowing that it can lead to such variable expression even in carriers, what does that inherent variability really tell us about the ultimate battle, if you will, between nature, the severity of the mutation itself, and cellular regulation, the inactivation process, in determining a person's actual health outcome?
That's a deep one. That spectrum of protection and vulnerability means the line between being simply a carrier and being truly affected is, well, it's actually dictated cell by cell, tissue by tissue. It's not always a clear boundary.
A battle fought at the cellular level. Something to think about.